Hello, and thank you for joining today's cutting edge conversation about the innovations addressing orphan and rare diseases, sponsored by OM1 and Cardinal Health. We appreciate your involvement. Our first speaker today is Dr. Joseph Zabinski, the Senior Director of AI and Precision Medicines at OM1. Our second speaker is Dr. Jonathan Kish, Senior Director of Real-World Evidence and Insights at Cardinal Health. Together, our speakers will discuss how new technologies and real-world evidence are driving research in precision medicine to treat rare and orphan diseases. Thank you both for joining me today. My name is Megan Loy, and I'm the Category Director for In Vivo Services at Scientist.com, as well as a board member of the North American 3Rs Collaborative. For the last 3 years, I've been working closely with our clients and suppliers to advance relevant categories, as well as offer animal welfare quality and compliance support. Previously, I spent the majority of my career at Covance Laboratories in Madison, Wisconsin, working both in the vivarium and in client services. I have a bachelor's degree in agricultural sciences and a master's in biotechnology. Before we get started, we have some brief notes to make your viewing experience better. First, if the panels on your screen are too big or too small, you can zoom in or zoom out in your internet browser to adjust their size. Next up, the webinar is being recorded, and the video and its associated resources will be available to watch shortly afterwards. We'd love to hear from you and invite you to share any questions, thoughts, or comments in the Ask a Question panel shaking on your screen. We will be taking your questions in a Q&A session after the presentation. If you'd like to learn more about the research or technology presented today, explore the resources panel, where we've added a few relevant links and downloads. And finally, if you happen to have a technical issue, refreshing the page will nearly always fix things. But if it doesn't, please reach out using the Ask a Question box and we'll help you troubleshoot. I will now begin with introducing you to rare and orphan diseases. An orphan or rare disease is a condition that only affects a few people. So in the US, we consider rare and orphan diseases to impact less than 200,000 patients. In the EU, we say it's fewer than 1 in 2,000 people in a general population. So it's actually a small amount. The grid here on the right side of the screen is actually 2,000 squares, and you can see there's one red one right in the middle. So a rare disease impacts less than the number of people in that red square. Now, there have been some advances in the US and abroad regarding rare and orphan diseases. So the definition as we know it was actually created by Congress for the Orphan Drug Act of 1983. And rare diseases became known as orphan diseases at this time because drug companies were not really interested in adopting them to develop new treatments. As you can see, there wouldn't be a lot of patient population. So the Orphan Drug Act really created financial incentives that encourage companies to develop new drugs for rare diseases. And the definition was needed, obviously, to establish and to encourage the conditions that would qualify for these new incentive programs. So other countries do have their own official definitions of rare disease, as I mentioned in the EU. And they actually have a regulation, 141 in 2000, that's the orphan regulation. It does something similar to the Orphan Drug Act in the United States. So what actually are some orphan diseases? Well, the fact that orphan diseases are rare, specific ones, doesn't mean there's not a lot of them. And it's actually estimated that there are as many as 8,000 rare diseases. Most estimates say 6,000 to 8,000. So Americans living with rare diseases is estimated at 25 to 30 million people in the population. And I have some examples for you here. There's some things you probably never heard of and some things that sound really intriguing, like watermelon stomach. You know, we might not know somebody individually with that specific condition, but it's likely that you know someone with a rare or orphan disease. And the estimates around it are actually really difficult to figure out because there are few types of these rare diseases that are actually tracked in people. So rare diseases can be infectious, they can be caused by birth defects, they can be cancers, they can be genetic conditions, and not a lot of those are tracked overall. So in the example here, you see there's one for kidneys called IgA nephropathy. I actually have IgA nephropathy, so I have a rare orphan disease. So if you didn't know someone who had one before, now you do. There's not a lot of treatments for it right now. There's currently some trials going on. And it's a really interesting thing to think about, that even though these individual diseases are so rare, the chances of you knowing somebody in a general population that has one of the 6 to 8,000 is actually quite a bit higher. Now, there are also lots of uh, people and places working on cures or treatments for these rare diseases. So the NIH in the US has a Genetic and Rare Disease Information Center, and they are doing a lot of research as well as trying to raise awareness about rare diseases. So Rare Disease Day is actually coming up really soon. It's the 28th of February this month. The FDA, as we mentioned before, had the Orphan Drug Act in 1983. And alongside that, the EMA, the European Medicines Association, also has their own regulation. The World Health Organization creates priority lists for these diseases and basically lists research needs so that researchers can target what's most critical. And then finally, the UN actually recently had a resolution on persons living with a rare disease. So in the next 20 years or so, they are really looking to advance research in these areas and help all of the people worldwide who are living with these types of things. So with that being said, now I would like to welcome Dr. Joseph Zabinski from OM1 to the stage. And Joseph, thank you for taking the time to speak with us today.
Great, thank you, Megan. Thank you for providing that quick overview of the situation facing a lot of people out there with rare diseases, and reminding us that even though any individual rare disease is rare, in aggregate they're quite common, and there's some things that we can do about that to help. So I know we don't have too much time together today, so I'll dive right into the materials I've prepared on the topic of using real-world data as well as artificial intelligence to better identify and understand rare disease patients. By way of background, as Megan said, my name is Joe Zabinski. I'm Senior Director of AI and Precision Medicine at OM1, which is a healthcare data and technology company where we focus on outcomes, including those affecting patients with rare diseases. And so just to set the stage, I'm sure that everyone here is probably familiar with both of these terms, real-world data and artificial intelligence, or at least you've probably heard them thrown around in a lot of presentations because there's been a lot of hype around these things for a number of years now. But I wanted to pause before diving into applications just to remind everyone of what these things are and how they can work together. So when we talk about real-world data in healthcare contexts, particularly when we're talking about drug development or real-world epidemiology studies, I always define real-world data as all those data that are not generated by clinical trials. So sort of any other data point that you can imagine that would reflect a patient's status or their journey that doesn't come from a clinical trial, I think is fair game to be considered real-world data. So that can include information from claims records, medical claims, pharmacy claims. It can include data that come directly from the electronic health record system, but it can also include things like socioeconomic status data, data around procedures, around consumer behavior, many other types of things. And at OM1, we've really made it our mission to gather all of those different data types for as many patients as we can and knit them together to create as strong and comprehensive a longitudinal trajectory for a patient as possible. Artificial intelligence is really a set of algorithms, a set of computer programs that take large data assets, look for patterns in those assets, and then associate those patterns with outcomes of interest. Of course, in healthcare, we're talking about things like, does someone have a disease? If someone has a disease, what are their risks of progression or of complication? Or how are they going to do on a particular treatment? We can also think about things like, will a particular treatment work in a condition? These are all applications of artificial intelligence. And these two tools can really work together by providing, from the real-world data side, a lot of rich data for training, testing, and validation to artificial intelligence algorithms. And then for the artificial intelligence algorithms to feed back into the real-world data assets through data augmentation and amplification, through basically strengthening what's in those real-world data assets. So just sort of high level, being aware of what these things are and knowing that they can work synergistically together. I want to talk a little bit about, sort of having set the stage that way, 3 areas where AI and real-world data can help in rare disease. The first of those being helping us to better understand understudied populations and rare disease phenotypes, then in enhancing real-world epidemiology through helping us to see true real world patient populations, not just those that sort of exist in prior databases or studies. And then finally, using these tools to actually look for patients in the real world to shorten time to diagnosis. So with respect to the first of those applications, one of the really interesting things to note about rare diseases that, again, I'm sure is very familiar to many in the audience, is a lot of times rare diseases live inside of common diseases, right? We can take sort of a large common condition that's not of particular surprise to anybody, but when we start looking inside of it, there may be small subgroups of patients who are really meaningfully distinct and need to be thought of and treated differently, but who can sometimes get sort of washed out into the broader disease classification. Um, an example here is lupus. Lupus is kind of right at that threshold that Megan was describing in terms of its epidemiology and prevalence of whether or not you would call it a rare disease as a category. But when you look into the real world data, and we start sort of from a starting point of 300 million patients, so we can usually take a look at anything at OM1, and in the area of lupus, we have quite a few patients we can start to look into. What we notice is that there are distinct manifestations of this disease that clustered patients as meaningful subgroups within the broadest category of lupus. And some of these are pretty well known and describe things like lupus nephritis. But there are others where you see sort of patients with similar manifestations or patterns that are less well described or less well focused on, both for clinical development as well as in treatment. With real world data, you can find those groups. You have the scale of data assets to say, rather than just looking at sort of one big group or a few, let's really parse this down meaningfully. And then we can use artificial intelligence to say, what does one subgroup of interest have in common? And then let's go kind of proactively look for that subgroup in other databases, in other care settings, other clinical applications. So sort of using the AI to understand trajectories of people who have a distinct rare condition or distinct rare phenotype of a more common condition, and then labeling patients at risk of having that phenotype is sort of one of these key advantages. Um, that sort of plays into the second highlight that I wanted to bring up here with respect to using real world data and AI in the area of rare disease. And that's around the idea of enhancing visibility into real world patient populations. So, it's hard to study rare diseases, as I'm sure many of you know, just because it's difficult to access sufficient patient numbers to really say things that are meaningful about the disease as a whole. And one of the challenges that comes from that starting point of difficulty is a lot of times the literature, sort of the medical knowledge, the training, they all focus on one manifestation or one sort of classic phenotypic presentation of the disease. So you might say, the classic presentation of this disease is in younger men who start out with some neuropathy or start out with some cardiac issues and then progress to X, Y, and Z, right? And that's not a bad description of a particular condition when we say, here's a classic description of it, but it can tend to leave out patients with many other types of presentations, many other types of ways that the disease can manifest that can have some overlap with that classic phenotype, but not completely. And when that sort of focus on a single classic phenotype happens, it can build on itself. A lot of evidence is generated about that phenotype. Epidemiologic studies are conducted on that phenotype. Ultimately, sometimes medications can be developed that are really targeted towards that phenotype, and it can leave out people on the edges. One of the nice things with real world data is it can show you where a lot of those edges are. And it can do that in a number of different ways. First of all, patients with demographic characteristics that look underrepresented in prior work. For example, in some conditions, most research has focused on men, but there are women with the condition, and sometimes their presentation of disease may be different. Real world data can help with understanding those differences better by saying, even though not as many women perhaps may be diagnosed as they should be, we at least have some, and we can compare them sort of apples to apples with the more classic type of the disease to see what we're missing. Another way to think about this is with some of these heterogeneous disease manifestations. Sometimes, if a patient doesn't check all the boxes on the checklist of a classic subphenotype of a disease, they may sort of be said to be variant or just be sort of difficult to understand a little bit, and maybe shunted off sort of onto the edge of our thinking. In some cases with rare disease, a patient who doesn't match the most classic diagnostic checklist may not even get diagnosed at all. And real world data can help us to see where patients with, some of that fringe overlap may be existing, and to understand that, maybe they constitute their own clusters, their own sort of groups, that are meaningful with respect to understanding the whole spectrum of the disease. And a lot of times with respect to treatments, treatments can help these patients too. They just may be getting overlooked. And then finally, this kind of real world epidemiology and visibility can happen at the level of geography as well. Sometimes patients are more known in areas where there are more physicians or other clinical resources who are looking for those patients. And so looking sort of at a broader geography and using that to find visibility into understudied or undiagnosed groups of patients geographically can be a big area of help here as well. The final area of application I'll just quickly touch on is this concept of patient identification or patient finding in rare disease. It's tough for clinicians to get to a diagnosis of rare disease because of that long tail effect that Megan was mentioning, because there's so many rare diseases out there, each of which has its own particular characteristics. And as a result, it often takes patients quite a long time to be diagnosed with the rare disease that they have, from the time that they first start manifesting symptoms until they get diagnosed. And that path usually involves lots of physicians, lots of tests, a lot of frustration, and sometimes treatments that are ineffective for the patient. Patient finding using a combination of real world data and AI can actually take advantage of some of that stepwise process and use it to make it easier for other patients. So we've built this tool called Patient Finder. It's an AI tool. And what it does is looks in our real world data assets at those trajectories. It observes, for example, this patient first goes to their primary care physician. That physician refers them to, this specialist. That specialist does this test. Then they go back to the primary care physician. We can sort of look at that patterning and sequencing information, and then look for the same pattern in other patient journeys that are manifesting in the real world data assets, but where those other patients haven't yet reached the end of their diagnostic journey. And by doing so, we can say these patients here, maybe a small group, are at much elevated risk of having this undiagnosed condition. We've done this successfully in rare disease. This is just one example of a rare lysosomal storage disorder that we've worked on, where we achieved really good predictive performance, but also really good concordance with the clinical view of what drives this disease's presentation and progression. And that's one of the key things when you're working with anything with artificial intelligence. You want it to be clear and understandable to clinical experts and to patients, so that you're not just saying the machine says this and that's sort of the end of the story. You want to be able to talk about why the machine is pointing out specific individuals as being at much elevated risk, particularly in rare disease, where any kind of information can be a little bit bracing and scary to be received by a patient. One of the nice things about these tools, I don't think this was really true if we had been having this conversation a few years ago, but it is starting to be true now, is these tools are starting to move from kind of existence just on the computer to real world deployment. And I think that that's particularly true, in the area of some of this patient identification work. We're certainly starting to move into clinical process where we say, we ingest large data sets, we isolate those patients who are most likely to have the rare condition we're looking for, and then we hand that information back to clinicians to say, if this makes sense to you, this patient can be contacted to see if they want to get a genetic test or other evaluation for the condition. So that kind of closed loop that results in real world diagnosis is very exciting, and it's possible now. So I'm just about out of time with my remarks, and I'll hand the ball off to Jonathan. But I just want to close by saying to everyone, real world data, all those data gathered outside of clinical trials, augmented with and kind of focused to personalization through AI, they're very powerful tools that can help us in rare disease, where often saying much of anything is tough because of the rareness of the conditions. And these tools are really coming into their own in this area and letting us see things and do things that weren't possible even a couple of years ago. So I'm very excited to keep working in this area. So thank you, everyone.
Thank you, Joe. That was really interesting. And I can tell you that My condition is most common in Caucasian and Asian males. So, as a Caucasian woman, they were a little bit surprised to see it. And it did take me about 8 years to get diagnosed. So, I can confirm what you're saying. So, these developments are really especially exciting for those of us that have rare disease. And with that, I'm so excited to welcome Dr. Jonathan Kish to the stage. He's going to begin our next presentation. So, welcome, Jonathan, and the floor is yours when you're ready.
Wonderful. Thank you for having me today and Cardinal Health and the opportunity to present. Today, we'll talk about rare patients and rare data, leveraging decentralized networks in cancer. Just by way of brief background, again, my name is Jonathan Kish. My background is in epidemiology. I've been with Cardinal Health for 6.5 years, leading our real-world evidence and insights program here. Prior to Cardinal Health, had some time at other consultancies and then some great learnings at the National Cancer Institute. And today we're going to talk specifically about that. So I'm going to focus my conversation on what is often thought of as common, but in certain cases can be very rare. We'll talk about rare patients and rare data actually in cancer. And I will move ahead to the next slide. So we'll talk about why I believe and why Cardinal Health is focused on developing networks and data assets in oncology, given the explosion in precision medicine. I'll go through some case studies about how real-world data can be applied in these patient populations in specific use cases. And then I'll talk a little bit about the networks that we've constructed to help support those use cases for generating real-world data and real-world evidence. So to begin, we all, I think, probably have some experience in either personally or in studying cancer. There are obviously numbers of different cancers that exist. And the important thing here is that while they, as an aggregate, as our speakers have alluded to, these cancers might not be considered rare or orphan, with the increased use of and development of therapeutic targets at the molecular level, these cancers have become more rare in that sense. So we can think of a number of different examples where the overall incidence or prevalence might be in the hundreds of thousands. But when we start to look at those patients with specific patterns of mutations and other biomarkers, that group becomes smaller, right? And we're starting to see more and more of that as we go along. I'll take one brief walk down memory lane here to give that example a little bit more detail. And so not more than 15 years ago, lung cancer was considered, basically one overarching tumor type that had two specific markers of interest, EGFR and KRAS. That since in the ensuing 15 years has dramatically changed. As little as 7 years ago, we had around 10 biomarkers that were specific to molecular aberrations, oncogenic driver mutations in lung cancer that have been identified and for which targeted agents had started to been developed. And as recently as 2 years ago, we can see that continues to change and explode in terms of our understanding of the patterns of oncogenesis and those driver mutations and how they all play together. And this leads to an interesting conundrum in a lot of ways. To Joe's point and others, we have an overall group of patients that we want to understand, but now we have rare patients, right? And we want to be able to understand the characteristics of those patients, who they are, where they are, what is happening to them. And then even more important would be their outcomes. In the drug development life cycle, we know that many phases of the development have now shifted a little bit earlier because of the rarity of some of these targets. And so historically, where the gold standard may have been a phase 3 randomized controlled trial, comparing an intervention, in two groups, right, to those randomized to receive it or not, now we're starting to see more and more of the drug development life cycle focused on phase 2 trials, where we may not have a comparator arm. It may be unethical to do that type of study. And in that case, real world data is increasingly useful to help contextualize or even compare the results of things that we see in the trials. When we have mutations and targets and prevalence of disease that are, 1 to 3% of the overall population of patients, being able to find those patients to participate in trials is problematic in having that work. So how have we solved for that? We'll move ahead and give some more case studies. So an example would be here on the screen, talking about the use cases for real world data. When we at Cardinal Health here, we're focused on how can we support driving and developing real world data and evidence that supports evidence-based medicine for the clinicians. So again, the example would be to understand how for non-small cell lung cancer patients, the use of a specific sequence of therapy in patients without a specific type of genetic marker could be compared to data from a real-world clinical trial. So in this case, the rarity starts to come in when we talk about non-small cell lung cancer, and then we focus on patients with squamous cell disease, and then we focus on patients who received first-line immuno-oncology therapy, such as pembrolizumab, and then we focus even on those patients who may have received a subsequent therapy, in this case, afatinib. How do we solve for that, right? How do we find these rare patients, in this case, who are exposed to a sequence of therapies and have certain specific markers? And so what we've done here at Cardinal is implement an approach to working with a large network of clinicians. We'll call it our decentralized network, and we'll talk more about that, to be able to identify those rare patients, relying upon those clinicians and their expertise to be able to find them and to give us the rare data that we also need to contextualize the results that we see. Similarly, the use case continues to evolve, right? We all seek to publish the data that and evidence that we generate to be able to support evidence-based medicine. As we know, as I mentioned, the evolution of the use of real-world data has come into the regulatory framework. And so in this case now, with FDA guidance and industry guidance being promulgated, we can use these real-world data, particularly in the case of rare and orphan diseases, to be able to use that in submissions to the agency, right? So we can collect these data, we can compare them to pivotal trials, we can use them as supporting documentation, again, to help the FDA and the agency make informed decisions to support regulatory approvals. And so here, Cardinal Health worked with MorphoSys several years ago now to identify relapsed refractory DLBCL patients, diffuse large B-cell lymphoma patients who had received a specific sequence of therapy. And the important thing to remember here is that it wasn't just those patients who had that diagnosis, but to inform the research and to be able to do the comparative effectiveness work that needed to be done, there were a set of covariates that needed to be matched on. And that included 9 different covariates of patient clinical characteristics. And so to be able to do that, you need a large sample to start with, right? You need to be able to find these patients who are rare to begin with, and then even rarer when we start thinking about their clinical conditions. And so in that case there, we used our decentralized network called Open, our Oncology Provider Extended Network, to be able to identify those patients and collect the data of interest in order to inform this research. So let me talk a little bit about our networks here to illustrate how they can support these types of rare patient findings and studies. So Cardinal Health has our long-standing network called Open, our Oncology Provider Extended Network. We also have built networks outside of oncology and rheumatology, neurology, and other rare diseases. And really what these networks are all about is targeting and finding providers who are treating these patients of interest. If you're specific to one data set, then you're going to have a limited footprint in that one area, right? There's obviously a lot of work going on to integrate data from across EMR systems, hospital systems, claims. Joe mentioned how we pull all this real world data together. But at the end of the day, it's important to be able to find those clinicians who are treating those patients. And so with that, Cardinal has created these networks where we leverage those physicians to be able to identify those patients and extract the information that we need across these different specialty therapeutic areas. The power of that network really lies in its representation across the US. Again, where you may be limited in some cases to patient finding or using real world data from one EMR system or one specific group or one hospital system, we've really embraced the idea of decentralization in this way. We know that we're going to need these providers and these practices to work with us to be able to find these patients. And so in oncology specifically, we've created a network that has over 500 unique practices that we can leverage at any one time to be able to find these rare patients as we move down the funnel from non-small cell lung cancer to ALK positive to specific sequences of therapy. I do want to talk a little bit more then about, not only rare patients, but rare data. And so it is the case that many of the things that we study in clinical trials aren't replicated in the real world. There's often for example, the need to study patient-reported outcomes, right? And those things aren't necessarily routinely collected in the real world in a structured format that is similar to that done in the trial. And so again, here where Cardinal Health is working to improve data understanding and the evidence that we can generate is to use those physicians to be able to work with us to find the data of interest. And what that means is when we talk about things like tumor response in oncology, where we don't have a standardized assessment like RECIST, the solid tumor assessment done in the real world setting, how can we find the data points that are necessary to support developing tools that can be similar, not necessarily to the same standard of those trials, but similar enough to provide robust evidence to contextualize the results of a clinical trial vis-a-vis real world data for these patients. And so it really takes that provider, it really takes that connection to that patient to be able to pull out those unique aspects and insights for the data. Did the patient discontinue therapy, for example, because of intercurrent illness, or was it due to disease progression, or was it due to toxicities? How do those three things play out? And that's a very difficult proposition to uncover when you're simply looking at the structured elements of a data set that you have. And so again, we rely upon that clinical expertise to be able to feed into our data sets to allow us to make those determinations based off of what the provider is reporting. We've had some success in this area, I don't mind saying today. And really, we believe that our approach to leveraging those physicians to augment a lot of that data allows us to drive better insights and better care for patients, especially in these rare conditions where not only is the disease rare, but also the data points of interest could be rare. Most recently, we began work to evaluate how in lymphomas, clinical endpoints in the real world can be viewed in the context of a randomized controlled trial. So working with the FDA, Cardinal Health will go out and identify those patients. We will identify those rare data points, and we'll look at the concordance between what is the tumor response outcome in the real world vis-a-vis what it was in the clinical trial. And so with that, I will begin to conclude and say that in rare patients, the evolution has begun to occur in terms of the use cases and what we need these data for, not only from, as Joe mentioned, how we can identify those patients earlier, but also at the end of the day, how we can improve outcomes by leveraging the data and insights that exist to drive better patient care. So with that, I'll stop and thank you for everyone's time.
Thank you so much, Jonathan. That was really interesting. And that concludes the presentation portion of our event. So thank you to the audience for following along and learning more about the advancements and the achievements of today. And we're really excited that we could all connect and talk about the challenges and opportunities that are available in this space. So we can now get to some of the questions that you've all asked. And feel free to continue entering your questions into the Ask a Question box, and we'll get to as many as we have time for. So the first one is, how can AI and real-world data be used in clinical development, for example, in trial design?
So I can take a quick stab at that first, and I'd be curious to hear Jonathan's thoughts, of course, as well. But with respect to trial design, I think that the point that was just being made about rare data is particularly useful when designing rare disease clinical trials, understanding how difficult it is to get data in in trial contexts, but even in real world contexts, is very useful before committing to a certain design, because different pieces of information and types of pieces of information can have very different levels of complexity to gather. Of course, there's always a question of what's going to be required for regulatory submission, but real world data can help understand what's out there and what may be usable in practice. The other aspect that occurs to me is in some of those areas where it may be unethical to do a placebo-controlled study, that's again a challenge that comes up with rare disease, and where with dealing with small n's, being able to do something synthetic using data that are available can be very helpful.
Yeah, I would
John,
agree. Sorry. Yes, I would certainly agree. I think that the promise of AI and the ability to leverage those large data sets in trial design and drug development is increasingly useful. The ability to be able to, as Joe alluded to, right, even simply identify whether those patients will exist in different types of data and different types of geographies or other areas allows us to really figure out how to effectively design a trial, and move the needle forward. So I think it's all very promising right now.
Thank you both. The next question is, can AI be used to identify potential uncertainties and the extent of uncertainty in clinical data?
I would say certainly, particularly if you're able to use AI on a large data set that can make smart comparisons, and particularly understand where gaps are by comparing a number of different representative samples of data, and then even predict where you may have the most trouble gathering data and getting a complete clinical data picture in the future. That's totally possible.
I would just add that I think it's a very relevant topic, particularly in oncology. As I mentioned, many primary endpoints in early phase trials are tumor response. We all know that there's been a lot of initiatives put forth to have not only cancer diagnosed based off of radiographic images and AI algorithms, but also to work towards using those algorithms to be able to remove that uncertainty when it comes to what is the patient's response to therapy. So just as a brief example, when we go out into the field and we work with our clinician network to obtain the patient's response to therapy, folks on the phone may be familiar with complete response or partial response and stable disease and some of these definitions. We often see that there's a lot of over-reporting of the response that a patient may have out in the community. And there's a lot of reasons for that, both good and bad, but at the same time, there's a lack of rigor in that, right? There's no standard that's applied. And so to the extent that the algorithms can be trained appropriately to be able to, remove some of that uncertainty, I think will go a long way in supporting, in oncology at least, the ability to use those data for comparative effectiveness when you're bringing in a real world cohort.
Excellent. Thank you both. Another question that we have is, are there any incentives or help for pharma industry to develop new drugs for rare diseases, like patent expiration date extension or others? And I can just refer back really quickly to the beginning of the presentation where I talked a little bit about the Orphan Drug Act. That actually does allow for some financial incentives as well as patent extensions. The European Act is similar to that. But Joe and Jonathan, if you want to add any detail to that, we'd be happy to hear you.
I don't have any specific detail to add.
Yeah, exactly.
Okay.
I would refer to our regulatory sciences division.
Exactly.
Okay, well, I apologize for throwing you under the bus there after I answered your question.
No, you just know more than us.
Oh gosh, I wouldn't go that far. What challenges can you run into when you work with ultra rare diseases, and where they're in situations where there are very, very few patients that are actually diagnosed?
Well, I'll start. if there are very few patients that are diagnosed, the number one challenge is going to be finding them, right? And finding them in the appropriate window of time necessary to be able to do the work that you want to do. I think it depends a lot upon the use case. I mean, the fundamental issue that as an epidemiologist, I always wrestle with in discussing with our clients is one of sample size in a lot of ways. So let's move beyond the idea of, trying to find these patients to improve their care for a second and talk a little bit about if we're doing research, right? Well, we need to have some accuracy and precision in what we're doing. And so there's going to be a limitation to that. I think most stakeholders understand the balance between statistical precision and clinical relevance in a lot of ways. And I ask all the folks who are on the phone to continue that discussion, right? That a p-value, as many of us probably learned, is one measure, but it's not the only measure, right? It does give us some of that insight that we need to have. So I think that's the fundamental challenge that we face is understanding when have we generated enough data, even though we may have these very, very rare patients, to be able to, move the needle, right? To actually improve the care for these patients overall.
Yeah, I would agree, certainly with all of that. And the question of sample size, as Jonathan points out, is always the big challenge with rare disease in general, but especially with ultra rare disease, sort of the rarest of the rare. On sort of an AI side of this, there's a number of things that pop into mind as interesting when working with ultra rare diseases. One of those is just that I found sometimes you just don't have enough patients to do much with an ultra rare disease, but you can look one level up in the taxonomic tree for that ultra rare disease and say, I may not be able to go, say something useful or find patients with the ultra rare condition, but maybe I can find patients in the class of conditions of which that ultra rare disease is a member. And maybe that's not useful, but maybe it is. Maybe it's enough to hand a clinical team patients who, with reasonable statistical certainty are in that broader class of disease, and then allow them to do the final kind of investigation to get down to the ultra rare disease level. Sometimes that's very helpful, and it's tractable from a data perspective. The other thing is just, I know part of the question was about conditions that just don't have that many patients diagnosed. Sometimes that's because the patients don't exist, and sometimes it's because there hasn't been a good way to diagnose the patients yet. I was just working with a team earlier this week on a condition that only received an ICD-10 diagnostic code in 2020. But since then, it's entered reasonable usage and has started generating real world records of patients with the condition. One of the nice things that you can then do is say, okay, now we have something that's like a gold standard cohort. It may not be very big, but we can at least use it to train an AI system to look deeper into longer term records and say, are there other patients that look the same, but just weren't able to be labeled because the coding didn't exist yet? That can be sort of I won't call it a low hanging fruit, but a medium hanging fruit for some of these applications.
Thank you. That's really easy or interesting to think about also how patients get diagnosed. For my condition, I had to have a kidney biopsy, and it was not a very pleasant experience, and it's not one that many people opt into unless they have to. So I can see why patients are not diagnosed for quite some time and why their data might not be available. They just haven't gone through with it yet. So speaking of which, how can patients leverage their data? How can they get it out there and work with their clinicians in order to improve their treatments?
Ooh, that's a great question, and one that we could probably do a whole separate conversation on. Most broadly,
good.
I don't think that the infrastructure is super well developed here yet. I think it will get there in the coming years, but I think patients having greater access to and autonomy over their data is kind of a new wave that we will see more and more of. At this stage, I would say, I would advise, what I try to tell people in my own life to do is to just be aware of your data and where they are when your provider says sign up for this portal so you can see your data. Like, do it if you can, and just take a look around and kind of be aware. And then perhaps when you go to a new provider, you can say, I've got something. I don't really know what's in it, or how to interpret most, but I at least know it's there. I think the awareness at the individual patient level is the place to start.
Fantastic. Thank you. Maybe we'll schedule another webinar to talk about that in detail. And then how do you approach rare conditions that don't have dedicated coding yet?
Well, from my end, I'm not sure if Jonathan, you have any other insights there. I already gave my answer, which is we try to look around the corners for them.
Yeah. Are there any other options
Right.
besides looking around the corner?
Well, I mean,
Sounds like that's
it's difficult to be sure. Joe's spot on with his response about looking around the corner. I think again, how you do that becomes the important aspect of it, whether it's through large data sets or whether it's speaking specifically with experts in that field. We have recent examples of that, just like Joe had, and probably talking to some of the same people in a lot of ways of trying to find a cohort for a real world arm that doesn't exist yet, right? It's not a condition that has a code. And so how do you build out your ability to look at that through EMR systems, right? And so you look for those component pieces and then have clinical input on that, right? If we collect all these data at the patient level about what we think are suspected cases, can we get to some agreement on those which are true cases in a lot of ways? And again, I think I'll go back to the idea of it's not a 100% perfect answer in a lot of ways, but there is a reason why we have qualitative research methods, why we do different approaches to different things to be able to come to some sort of agreement to at least take 2 steps forward, even if we sometimes have to take 1 step back as well.
perfect. Thank you both so much. And I think that's the end of our questions. So thank you so much for joining today's presentation, and a big thanks to OM1 and Cardinal Health for sponsoring the first cutting edge conversation of 2022. We now invite you to take a moment to share your feedback on the survey, and we would really appreciate your input. If you still have a question, it's not too late. Feel free to send it in now, and we'll forward it along to the speakers following the event. If you're interested in learning more about the research, we invite you to check out the resources panel. And if you're interested in seeing what's coming up next in the cutting conversations, you should click the register button on the side to find out more. In closing, we hope you enjoyed this scientist.com webinar, and we'll see you next time.